successful in screening and developing the organisms at the laboratory level for a very good industrial production, the overall process has not been successfully implemented at industrial level because the pilot plant operations have suggested that this would not result into an economically viable operation, and therefore such projects have been dropped. So the data which are obtained at this level, at the pilot level, always help in deciding if one has to progress further at the higher scale of industrial application of the microorganisms. In case of an encouraging response at the pilot plant, it is helpful in designing and constructing a full size operation. If the pilot plant suggests that the successful implementation of this process is possible at the industrial level, then as I said in the earlier part of this lecture, that in many cases it so happens that the corrections have to be made, some kind of modifications have to be made, some kind of further optimization has to be carried out at the pilot plant level. So such factors have to be taken into consideration, and all these has to be considered when we transfer this overall technology to the industrial level. So the information regarding the optimization, standardization of the conditions are to be very religiously transferred to the industrial scale operation. A pilot plant is always designed and constructed with provisions for process and environmental controls, and also it has to be always taken into consideration that industrial level plant is, as I said earlier, as big as a 3 to 5 storied building, and it requires different types of cleaning apparatus, the system of sterilization, and so many different parameters which are all to be standardized, which are all to be understood and have to be standardized before we jump to the higher scale, that is the industrial scale. એટલે મેં અગાઉ કહ્યું તેમ આપણે જ્યારે ક્વોન્ટમ લીપ મારીએ, જ્યારે એટલો બધો મોટો કૂદકો મારીએ તે પહેલાં આ બધી જ પરિસ્થિતિઓ આપણને સાનુકૂળ થવાની છે, અને ઇન્ડસ્ટ્રીયલ લેવલે આ બધામાંથી આપણને જોઈએ છે તે પ્રકારનું જ પરિણામ મળવાનું છે કે જે પ્રોડક્ટ છે તે તો સારામાં સારી ક્વોલિટીની હોવી જ જોઈએ, પણ એની સિવાય એ પ્રોડક્ટની શેલ્ફ લાઈફ, એ પ્રોડક્ટની વાયબિલિટી, એ પ્રોડક્ટની સાથે એસોસિએટેડ ઘણા બધા પ્રોબ્લેમ્સ, એ બધા જ પ્રોબ્લેમ્સનું સ્ટાન્ડર્ડાઇઝેશન આપણે પાયલટ લેવલે કરી દેતા હોઈએ છીએ, અને એ આખી સ્ટાન્ડર્ડાઇઝ કરી દીધા પછીની જે બાકીની બાબતો છે કે એ પ્રોડક્ટ છે એનું પેકેજિંગ કેવી રીતે થશે, એનું માર્કેટિંગ કેવી રીતે થશે, એનું પણ એક આખી જુદી એક સિસ્ટમ ત્યાં ડેવલપ થયેલી જ હોય છે. એ પર્સોનેલ એ બધી પણ બાબતોની ચર્ચા સાયમલ્ટેનિયસલી એ જ સમયના ગાળામાં કરતા હોય છે, અને ત્યાર પછી આપણે હાયર સ્કેલ ઉપર જઈ અને ઇન્ડસ્ટ્રીયલ પ્રોડક્શન કરતા હોઈએ છીએ, જેની વિશે સ્નેહા હવે વાત કરશે આપણે.
So we have reached up to the pilot scale, and the things have already been elaborated in the required detail. So I hope all of you must have the clear-cut idea about the scale-up, how the studies are carried out, how the different parameters are studied at different levels, laboratory level, pilot scale level. Pilot scale studies are carried out considering not only the product, formation of the product, of course in comparatively smaller quantity, and then the product as well as the process itself, they are assessed, they are evaluated for different criteria before finally the product is ready to be introduced into the market for sale. Now this slide shows you the steps in the process of scale up, where it has already been said that for any industrial fermentation process, the economics of the process is very important. You know, there are many basic factors that influence the overall economics, and we have to see that all these parameters are kept in check then and then only the economic viability of the project is possible. And these also includes the market size and the competitive selling. And when all these informations are made available during the pilot scale studies, then they provide good guideline for setting the limits of manufacturing costs. So considering all these points, finally one may have the idea about the manufacturing costs of the products, and one may also come to know about where the substantial saving on the cost can be made. Now, as it has been said earlier that laboratory studies and scale up planning should be conducted simultaneously. That is what we have to think about, the laboratory scale studies as well as the scale up planning side by side, so that it would be possible to reduce the problems that might occur during the scale up of the fermentation process. Now, as you can see here, this slide, which is titled definition, some of you might have idea that after covering so many things about the process of scale up, now the definition comes. You may find it placed at the wrong stage, but the idea of keeping this slide at this particular point was that you should be knowing in general about the scale up process. We often talk about the laboratory studies and pilot scale studies, but many of you might not have the clear idea about what are the pilot scale studies, and whatever information we get at the end of the pilot scale studies, how they are actually helpful, how they work as the guideline for the same process to be conducted at the final level, commercial level. So after creating the required background about the scale up process, about the pilot scale studies, we have thought that now let us give the definition of the process of the scale up. As you can see in the figure that has been kept side by, it starts with the laboratory scale. That is the, if you remember the very first slide, which was giving the overall idea of the scale up process, the process starts from the stock culture. It might be there in the form of a slant culture or a petri plate culture, and then gradually the quantity of the medium, volume of the medium and the size of the unit increases, and finally the process ends where it was shown that the product was transported, it was kept in the truck for sending it into the market. So here the figure shows it starts from the laboratory scale, pilot scale, once again still higher scale, that is the demonstration of the pilot scale projects, and then finally the commercial scale project is there. So now we can define the process of the scale up as it is the determination of proper operational conditions. Operational conditions, I hope all of you understand, that is the conditions which are being operated, which are being implemented, which are being used at the commercial scale, and these operational conditions are based on the information that has been obtained from various small sized tanks that have been used during the pilot scale studies. As I have already told you that even the pilot scale studies are carried out using several small sized fermenters, and by end of such studies, whatever information we get, it is hoped that the same conditions, the same operational conditions will be suitable for or could be successfully employed at the large scale in order to get the required quality and quantity of the product in the defined period of time. So that was about the scale up, different levels of the studies, and now let us have some information about the basic problems or the scale up problems which are very frequently encountered during the scale up process. So whenever we say it is scale up, it immediately comes to our mind that it is what are the immediate changes that are observed. These are the two immediate changes are the size of the fermenter and the volume of the medium. As it has already been said that as we go further from laboratory to pilot and then to the grand scale, we are gradually increasing the quantity, volume of the medium used for the studies, and so the volume of the medium is the one thing that is immediately changed or that is immediately affected, and the another thing is the size of the unit, that is the fermenter, which is used at the different level. And because of this increase in the size, increase in the volume of the medium and increase in the size of the fermenter, different problems may arise at this stage, and this is because the process parameters, as you know that all the individual fermentation processes are having their own standard parameters to be employed. For example, temperature, pH, supply of oxygen, agitation rate, dissolved oxygen concentration, etc. So all these factors are affected by these two changes, immediate changes in the volume of the medium and size of the fermenter, and so these types of the problems, these parameters have to be attended very carefully so that it is possible to find out some solutions so that the successful scale up is possible. Now, while going for this, that is while scaling up the process, sometimes negative results are obtained in terms of decreased yield, which is not desirable. So as you have already been explained very well that irrespective of the scale, whether it is the process is being operated at small scale or at the large scale, it is always desired that the yield should be obtained in its expected magnitude, in its expected quantity, that is the magnitude of the process is increased, but it should not be accompanied with the decreased yield. So if the decrease in the yield is found, in that case one has to find out what are the causes, what are the reasons why the yield has been decreased, and after studying the different parameters it would be possible to identify, to recognize the one which is responsible for the decreased yield. And after identifying that parameter, it is possible to rectify it so that the process can be successfully scaled up. So the problems that have been very frequently observed, they fall into the three main categories as has been shown in the slide. The major problems fall into three main categories. So all these categories, all these problems which have been described here, they are scale dependent parameters or the scale dependent steps, we can say. The first one, as has been mentioned, it is inoculum development. We know that all fermentation processes, they require healthy inoculum, active inoculum, and that too in sufficient quantity. It is possible that for a laboratory scale process, inoculum would be required in the quantity of few ml of the culture suspension, but will not remain same when the process is studied at pilot scale and when finally it is conducted at grand scale. You know that inoculum, quantity of the inoculum is usually there in the range of say 3 to 10% volume by volume of the medium used. So the first stage that is affected, the first parameter that is affected is inoculum development. So the point is, as the scale changes, as we go from pilot scale to the higher scale, it would be necessary to add some extra stages in the inoculum development program. You know very well that the development of inoculum is also specially studied. There are standard protocols for the development of the inoculum, depending upon the process, depending upon the process organism, it has to be produced, the inoculum, final inoculum has to be produced following that standard protocol. And so with the rise in the scale, it would be necessary to add some extra stages in the inoculum development program, so that finally we get enough quantity of the inoculum. Second one is the medium sterilization. It is also scale dependent parameter, and we know that the fermentation media are sterilized in order to reduce the number of the contaminating organisms at some standard which has been previously set, and irrespective of the scale of the operation, that remains same. So when the scale of the process changes, it would be necessary to reconsider the del factor, the unit that is being used for considering the sterilization of the medium, has to be reconsidered and has to be recalculated. And one more parameter, which is the environmental conditions, because when the scale changes, the organisms are exposed to a changed environment. So as here it has been elaborated slightly, while increasing or while adding the extra stages required, there is always risk for the culture to undergo degeneration, because you know that by repeating subcultures there is always a risk of culture degeneration. The properties might be changed, might be altered, and that would also be accompanied by the change in the physiological status of the organism at the time of inoculation. That is when the organisms are transferred, or when the organisms are actually inoculated into the production fermenters, their physiological status is very important. It is desired that they should be there in their active phase of metabolism. So by increasing the stages, there also remains a risk of a change in the metabolic status, physiological status, and that might affect the process negatively. In case of medium sterilization, it is also scale dependent factors, and sterilization regime, as it has been shown, regime, you know that it is the time and temperature relationship that has to be standardized for the sterilization of the